The Biochemical Society and Portland Press are pleased to welcome you to this webinar, part of the Biochemistry Focus webinar series. Topics in the series will include different research areas in the molecular biosciences, as well as practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers to feature in our webinar series, so please see the website for more details. My name is Stefan Roberts and I'm Honorary Meeting Secretary of the Biochemical Society. Our invited speaker today is Professor Maria Grazia Spillantini, the 2020 Thudichum Medal recipient, which is awarded to honour outstanding contributions to neuroscience and related subjects. Maria Grazia is Professor of Molecular Neurology in the Clinical School at the University of Cambridge. Her group works on the molecular neuropathology of diseases characterized by tau and alpha-synuclein aggregate. With her collaborators, Maria Grazia identified alpha-synuclein as the major component of the filaments that form the Lewy bodies in Parkinson's disease and dementia. She described one of the first mutations in the tau gene leading to frontotemporal dementia and Parkinsonism linked to chromosome 17, for which she was awarded the Potemkin Prize of the American Academy of Neurology in the year 2000. She's been elected a Fellow of the Academy of Medical Sciences and a Fellow of the Royal Society. Maria Grazia is currently Professor Fellow at Clare Hall and a Life Member of Peterhouse, Cambridge. In 2018, she was made Knight Official of the Star of Italy by the President of the Italian Republic. In this webinar, Maria Grazia will give her Thudichum Medal lecture entitled Alpha-synuclein, a central player in neurodegenerative diseases. Questions will be asked at the end of the webinar, but you can send in your questions at any time during the talk. So if you have a question, please type it in the questions box in the image on the screen. And I'll now hand over to Maria Grazia for her Thudichum Medal lecture.
Okay, first of all, I would like to thank the Biochemical Society for having selected me for this prestigious lecture and medal, and also Professor Roberts for the kind introduction. What I would like to show in my talk is to talk in particular of alpha-synuclein, a protein that was first described as such in the end of the 1990s, and what it is known and how we think it could be involved in neurodegeneration. So many neurodegenerative diseases such as Alzheimer's disease, Pick disease, Parkinson disease, and also motor neuron disease, among the others, are characterized by the presence of protein aggregates in different types of neuronal cells. And these protein aggregates made of normal protein in the brain that normally are in a soluble state, they at some point start to aggregate and to form these inclusions that have been associated with the appearance of the disease. In alpha-synuclein, in Parkinson disease, the characteristic clinical feature consists of rigidity, resting tremor, bradykinesia, and postural instability. These are the motor features that appear in a patient, and this is the characteristic posture of the patient. But when these motor features appear, the disease has already gone on for quite a long time, and indeed, the symptoms appear only when about 50% of neurons, dopaminergic neurons in this area of the human brain, known as substantia nigra because of the black pigment formed by melanin, are dead. And this is how it looks like in substantia nigra in a Parkinson case compared to a control brain, a brain from a controlled subject. But what is interesting is that it was described in 1912 that the neurons that are remained in this substantia nigra, they contain these eosinophilic inclusions. That, as you can see in this neuron containing melanin in black, they appear round. These inclusions are known as Lewy bodies, and this is because they were first described by Friedrich Lewy in 1912. he didn't really describe them in the substantia nigra. That was done by Tetriakov, but in the basal forebrain. And you can see that in these different cell types that do not have melanin, the appearance of these inclusions, they have a different shape from the characteristic round deposits. And however, Tetriakov decides to call these inclusions, both the round and the different one, Lewy body, because after the name of Friedrich Lewy, because he was the first one to describe. Later on, during the years, already in 1960s, there were several studies, and even before, the Lewy body were further studied, and instead of using only hematoxin and eosin staining, they were observed with electron microscopy. And this is the so-called typical Lewy body in the substantia nigra of a patient with Parkinson's disease observed by Lizia Forno using electron microscopy. This is considered a sort of a classical Lewy body, and it contains an array of filaments in the halo and then granular materia and filaments in the so-called core. This is associated, as you can see here, with lipids and then many organelles that are somehow entrapped into the filaments array of the halo. So nobody knew what these filaments were made of and what was the relevance for the disease. What was the relevance of this Lewy body for Parkinson's disease? Although these are sort of cytoplasmic inclusions in the substantia nigra, and then with these aggregates, they are likely to entrap numerous proteins that traffic through the cytoplasm. And therefore, about 150 proteins, if not more, have been described in the Lewy body. But it was not known what the filament consists of and what was the protein making them. The presence of Lewy body is characteristic of Parkinson's disease, and indeed all cases of sporadic Parkinson's disease, they have Lewy body by definition. So if some cases do not have Lewy body but still they have a Parkinsonism, normally they are not defined as Parkinson's disease. And several familial forms of Parkinson's disease, they also are characterized by the presence of a Lewy body. So how alpha-synuclein now we know is linked to Parkinson's disease, but how that happened. So in the 1990s, 97, when I was 94 actually, when I was working in the laboratory with Michel Godert and Ross Jakes, that was a research assistant, a biochemist, we were mainly working on tau protein and try to determine the tau isoform, tau phosphorylation, and we found an antibody for tau that was cross-reacting with something else. And we observed these two bands in a gel that were not tau. So this started as a side project in the lab. And Ross, that was very much involved in determining what these proteins were, they called them perfectin and imperfectin because the perfectin was a protein that any assay you were doing, any test you were doing was working well. Imperfectin instead was working less well. So we, Michel, they cloned the alpha-synuclein, these two proteins, and they were highly homologous. So at the end, they were called, they were similar also to another protein just described in those years, just here called non-amyloid plaque component by Tsunao Saito in San Diego. The thing is that he called this protein non-amyloid component because using antibodies, they had stained brains from Alzheimer's patients, and this antibody recognized amyloid plaques. And therefore, this was the name. But looking in the database, it was clear that the protein we had identified, and one of them was similar or the same than the one described by Saito, they were homologous to a protein described by Marteau and Scheller, and that's in Torpedo californica, and that they called synuclein because it was present in the nucleus and in the synapse. So the decision was taken to call these two proteins synucleins, and therefore the perfecting became alpha-synuclein and imperfecting became beta-synuclein. And these were the first two proteins described in a human brain with the name of synucleins. These are very characteristic proteins which and have a C-terminal part containing many repeat regions. There are 7 in synuclein, alpha-synuclein. They are consisting of a repeat that is KTKGV. And this repeats is very similar to what can be observed in apolipoproteins. So it is clear that from this presence that these proteins, they were able to bind to lipid membranes. As you can see, beta-synuclein has a smaller number of repeats, and the two proteins differ in this C-terminal part. What is interesting that beta-synuclein does not contain the so-called NAC region that was homologous to beta-amyloid. And therefore, this is another difference that between alpha and beta-synuclein. Later on, another protein known as gamma-synuclein was first described in brain in association with breast cancer. Indeed, more highly is metastatic is the breast cancer, higher are the level of this protein known as breast cancer associated gene 1, and it was described by G in the States. And this gamma synuclein, differently from the alpha and the beta, is more present in the peripheral nervous system than in the central nervous system. So we had antibodies specific for all these proteins, for the C-terminus, the amino terminus, and that could distinguish between the alpha and the beta-synuclein. And by staining in the brain of a human brain, like shown by Saito earlier, the staining was characteristic of a synaptic terminal, in particular presynaptic staining was observed. So what is the function of alpha-synuclein? Alpha-synuclein is one of the most abundant protein in the brain. And as I mentioned, it is involved in lipid binding, and it being located mainly in the presynaptic terminals in the brain, and it is involved in transport of movement of synaptic vesicles. And it is also involved in endocytosis, as is shown by Shriganga Chandra, and then by Sudov, that is involved in movement of the vesicle, and that has been reported that can be in the conformation of an octamer or a cup that for the cluttering vesicles. But what we also know that many other functions have been implicated for the protein, such as is a chaperone protein is involved in increase and decrease of that molecule. It is involved in synaptic plasticity, and also in the size of the synaptic terminals, in that when you knock out all 3 synuclein, and so there is no redundancy, we have very, very small presynaptic terminals, synapse, while when you have an increase, 3 copies of alpha-synuclein, for example, you have very large synaptic terminals. So the more recently, other functions have been attributed to alpha-synuclein because it has been found in plasma and in other organs, and therefore it seems it is also involved in inflammation, but this remains still to be investigated. What we know for sure that it is involved in binding to lipid membranes. Another function that we identified by serendipity was that we used to use mice that were knocked out for alpha-synuclein due to spontaneous deletion of the gene. And what we found when we added endogenous alpha-synuclein to these mice was that there was an increase of dopaminergic neurons in the substantia nigra, pars compacta. This was very surprising because we thought that perhaps synuclein, we didn't expect an effect like that, And this amount that is up to 13–14% increase, it is proportional to the dose, to the amount of endogenous alpha-synuclein. So we looked at the mice when they were adult, but then we decided to look also in the embryonic stage of the mice during development. And indeed, what we found was that at E10.5, there was no difference between wild-type mice and mice knockout for alpha-synuclein in the amount of Th neurons. But as soon as the alpha-synuclein has to start to be expressed, or is expected to be expressed around E13, clearly there is a decrease in the number of Th neurons in the substantia nigra compared to controls. And you can see here also that this difference, E13, is proportional to the amount of endogenous alpha-synuclein. So this effect is developmental. We don't know if alpha-synuclein is involved in the differentiation or in the survival of some dopaminergic neurons. But recently, Vladimir Buchman, who previously made knockout mice for alpha-synuclein and more recently has published in Neurobiology of Aging a nice paper where also there is an inducible knockout for alpha-synuclein, he clearly showed that if you knock out alpha-synuclein in adult mice, there is no change in the number of dopaminergic neurons, but this instead is reproducible during development. So the function of alpha-synuclein that somebody has reported to be related to Nurr1 or so on, during development still remain to be investigated. Then, as I said, we had alpha-synuclein, beta-synuclein, and they were cloned. And so we decided also to determine the chromosomal localization of this protein, the genes. And this journal, Genomics, they also asked us to name the gene. And so the gene was named for alpha-synuclein, we name it SNCA, and for beta-synuclein, SNCB. And they were located in chromosome 4q21 and 5q35 respectively. So when a year later, the genetic mapping of a risk factor for Parkinson's disease was located in chromosome 4q21 and 23, we were quite excited because it indicates that potentially alpha-synuclein could be involved because it was in this area. However, we were not working exactly on Parkinson's disease. We had no DNA, no brain. And so it was really interesting when the following year, this paper came up in 1997 that described the first mutation in causing Parkinson's disease, the first genetic mutation, because until then the gene was not, Parkinson's disease was not considered familiar. This alpha-synuclein, this mutation in the alpha-synuclein gene was identified in 5 Greek family and 1 very large Italo-American family. And the mutation was an alanine to threonine in position 53, so in the amino-terminal part of the protein. This mutation is quite rare. There are very, very few families. But also the thing was that the threonine is the normal amino acid in rodent. So there were some doubts if this mutation could really be pathological because of this presence in rodents. And it was a second mutation, an alanine in position 30 to proline, a few years later, that definitely confirmed the involvement of alpha-synuclein in as a gene involving familial form of Parkinson's disease. However, when we saw this, we had all the tools to investigate alpha-synuclein in the Parkinson brain. And so what we did, we look at Parkinson brain, some section from the Cambridge Brain Bank, and this indeed is the first Lewy body, this structure that I could observe under the microscope, but we did not have much tissue. And so at this point, Virginia Lee and John Trojanowski contributed, sending us tissue from dementia with Lewy body and from Parkinson's disease from that they had available in Philadelphia. And so putting together, it came out that we found consistently that Lewy body in tissue from Parkinson's disease or with dementia with Lewy body, they contained alpha, they were stained for alpha-synuclein. The problem, as I mentioned, is that there are many proteins described in the Lewy body. So what we really needed was to prove that these filaments that we had observed or had been shown in the Lewy body by Forno, by other Terry and other people, they were really containing alpha-synuclein. So, for example, this is the staining for alpha-synuclein that we obtain in dementia with Lewy body. This is a multiple system atrophy where the accumulation of eosinophilic structure is in oligodendrocytes, and this is also the staining in pure autonomic failure. So what we did, we extracted from these diseases filaments like we had done before for tau, and we immunogold labeled them with the help also of Tony Crowder and Michelle, and we could see that they were stained. These are the black of the gold particles entirely by an antibody for this, recognizing the C-terminal of alpha-synuclein, while only one tip of the filament was labeled when using an amino terminal antibody. And this pattern was similar in multiple system atrophy, in Parkinson's disease, in filament extract from dementia with Lewy body, but also in filaments made with recombinant alpha-synuclein shaken at 37 degrees. You can see that the pattern of labeling is exactly the same. And also this staining was different and did not recognize peridelical filaments formed of tau that sometimes you can find in the preparation, for example, of dementia with Lewy body. So clearly this indicates that the component of the filaments and then of the Lewy body described in 1912 by Friedrich Lewy, they were containing alpha-synuclein. And therefore, these diseases here, idiopathic Parkinson's disease, dementia with Lewy body, REM sleep disorder, multiple system atrophy, and so on, now they're also known as alpha-synucleinopathy because the presence of alpha-synuclein genetic mutation as the cause of Alzheimer's, Parkinson's disease, and the presence of aggregates of alpha-synuclein in Lewy body that are present in all alpha-synucleinopathy clearly indicate that this protein was relevant in causing and in the progression of the disease, of Parkinson's disease. So until then, as I mentioned, alpha-synuclein and Lewy body were recognized by eosine or by ubiquitin. Ubiquitin was the standard staining until the 1997. And what happened, however, ubiquitin was present also in other protein aggregates like neurofibrillary tangles, like amyloid plaques, and so on. So it was really quite difficult to distinguish by staining a brain of Alzheimer, for example, what was Lewy body, what was neurofibrillary tangle, and so on. Using the alpha-synuclein staining and comparing with alpha-synuclein, we could see that in multiple system atrophy, about 25% glia cytoplasmic inclusion could be revealed. For example, this one indicated by the arrow, they are negative for ubiquitin, but they are positive for alpha-synuclein. Again, if you look in Parkinson's disease, you can find the big Lewy body in some cells, and then positive for both synuclein and ubiquitin, but others that are not. And then it was possible to observe with alpha-synuclein antibody also these inclusions that are recognized now like Lewy body in Alzheimer's disease. And these are, for example, in the cortex in pyramidal cells. Therefore, using alpha-synuclein as enlarged the number of the list of diseases where alpha-synuclein Lewy body can be present. And for example, about 60% of cases of Alzheimer's disease, they also show now, we know, alpha-synuclein aggregates. The other interesting thing is that the staining with alpha-synuclein has allowed to study the progression in Lewy body formation. And this very nice work by Kui Sisto and Laura Parkkinen and Irina Rafuzov shows how in human brain shows that initially alpha-synuclein forms dot-like structure in the cytoplasm in early cases, and this dot they coalesce and then they form these sort of large deposits that are also known as pale bodies before forming the characteristic Lewy body. And this is a human brain where in a section of the substantia nigra, you can observe all these different type of alpha-synuclein inclusions. But in particular, you can see that the Lewy body described also in this case, even, sorry, before Forno by Duffy and Tennyson, they contain these aggregates, but they do not have a clear core, and this could correspond to the so-called pale body, where there are shorter filaments, more dispersed, compared to the dense Lewy body that we had observed before. And this is very important to consider when we look now with more sophisticated technique, what is the composition and the shape of the Lewy body, these different forms of aggregation. So the staining of alpha-synuclein and with alpha-synuclein was also used by Heiko Brack and Kelly Del Tredici to look at the distribution of Lewy body in the body, not only in the brain of people that had Parkinson's disease, and different or also controls. They look at several thousands of people, and what they observed that Lewy body could be found not only in the brain, but also in the plexuses surrounding the gut. And then the hypothesis came by Brack that it is possible that these alpha-synuclein aggregates in the gut, which could be induced by some environmental factors that happen in the gut, and or food or pesticide or so on, and then from the gut through the vagus nerve, these aggregates go to the brain, and then they start the formation of these aggregates. So this was a theory until, however, it became clear that there is a possibility of spreading of alpha-synuclein from one cell to another. And this work done, for example, by Patrick Brunding and Lee in 2008 show that, and confirmed also published by Cordova, show that in transplant, in the transplant that were used in Parkinson patients to compensate for the loss of dopamine, these transplants were from midbrain of fetuses. So they were really very, very young. They would have had the neuron 14 years or so on. But then they start to present Lewy body-like inclusions. The only possibility was that these inclusions were coming from the surrounding brain of the patients that contain a lot of alpha-synuclein aggregates. And now we know that indeed many people have produced animal model. They have investigated this spreading of alpha-synuclein pathology from one neuron to the other. And what we can see is that indeed it is possible that the theory of Brack of spreading from the gut, from the periphery to the brain, or also the olfactory mucosa, through the brainstem and nuclei, then reach the cortex, it is a real possibility. And by investigating this, it has been possible to identify several new symptoms of Parkinson's disease that precede the motor symptoms. And so, for example, it is about in 85–80% of cases with Parkinson, they had at some time in their life, 20 years before the motor symptom, constipation or olfactory deficit, but in particular, REM sleep behavior disorder. And so sleeping, these people, they act like they were awake during their dreams while they sleep. And this is because some of the nuclei that are related to sleep, they are affected by the alpha-synuclein aggregates. And this REM sleep disorder, in about 5 to 10 years, 95% of people seems to change to Parkinson disease, to convert to Parkinson disease. And the REM sleep behavior disorder is now also considered a sort of like MCI, mild cognitive impairment, is for Parkinson disease. And so all this has indicated that there can be some Lewy body in the brain, but when the amount become consistent in particular area like the substantia nigra, and they are associated with neuronal death, you have Parkinson disease. But if they spread to the brain, they can cause also dementia, and also they can cause dementia with Lewy body beside Parkinson dementia. So knowing all this, we want really to try to understand, to have a model to understand what happened in the brain when alpha-synuclein start to aggregate. And so we decided to make mouse models. And what we want was to have a mouse where there was loss of dopamine release in the striatum, loss of neurons in the substantia nigra parts compacta, dopaminergic neurons, total reduction of striatal dopamine in and then beside the release. But all these, there were models doing that did something similar, pharmacological model like MPTP or 6-hydroxydopamine. But what we want was that this pathology had to be associated with alpha-synuclein expression and aggregation. So we used a shorter, a truncated form of alpha-synuclein that we knew, with work with Michel Godert and Tony Crowder, that it aggregates much faster. And that's what you want in a mouse that live for such a short time. And so in these mice, for example, a model that we have published quite recently, we find that at 1.5 months of age, you have this increase in the presence of alpha-synuclein aggregation. This is 2 months of age, and in the substantia nigra, and this compared to a wild-type mouse of the same age. In these mice, there is no endogenous synuclein, there is only the transgenic. And you can see that at 1.5 months, you can obtain these cells full of dots, like I've shown you before, in the progression of alpha-synuclein aggregation. And then at 12 months, you have these very dense aggregates in the cells. In mice, there is no melanin, and therefore there is not possible probably to have these nice round aggregates like we find in dopamine, in melanin neurons in human substantia nigra. So then we look at the synapse, where alpha-synuclein is known to be most active. And what we found was we looked at other proteins that we know are involved in release of dopamine, such as syntaxin or SNAP25 or MOG13, but we could not find biochemically any difference. So we thought initially, well, that's okay, there is no change. But what we observed, and this was really consistently, and we had a lot of discussion with Pablo Garcia Reitbock when he was in the lab, was that in the staining in these transgenic mice looked very different from controls. You saw these little dots that we thought were clumps, that were like rubbish, a section badly cut, but actually they were always present. And they were also present for the when the staining was for VAMP2. So it became clear that perhaps there was an association between this clump and the other protein, VAMP2, SNAP25, and syntaxin. And indeed, there is a redistribution. So the amount does not change, but it is the redistribution of this protein involved in vesicle transport and neurotransmitter release. So we look at human patients, brain of patients with Parkinson disease with a very short duration of the disease, because we believe these are probably early effects. And we could find in the putamen and in the striatum that indeed the appearance of the staining for alpha-synuclein or syntaxin was quite different, and much more patchy than what was in the control brain, like we had observed in the mice. So we did double staining, immunofluorescence, and indeed the alpha-synuclein clumps, they co-localize with the clumps of SNAP25, syntaxin, and also instead for that, there was a large co-localization, but some molecule of that, they did not fully co-localize with the alpha-synuclein. And this was the staining in normal brain. So looking, for example, at the aggregates in the striatum using a different technology, using this term super resolution, we can find that the aggregates of synuclein that are in red, they increase in the presynaptic termina over time. And you can see that become larger, and this corresponds to an increase in insolubility of a synuclein and the appearance of larger bands of higher molecular weight. And this is double staining for VAMP2 and alpha-synuclein showing the presynaptic localization. In these mice, we went and looked at the dopamine content in the striatum, and we found that at 12 months of age, there is a clear reduction, significant reduction in the level of total dopamine in the striatum. We look then at the release of dopamine following potassium injection, so inducing the release of dopamine, and we use microdialysis. We observed that at 3 months of age, the release of dopamine was the same in wild type and in transgenic mice, but at 6 months of age, already there was a decrease in the release of dopamine in transgenic mice compared to controls. This was further reduced at 9 months and then at 12 months. So there was a progression in the alpha-synuclein accumulation, but which corresponded also in a reduction in the dopamine release. We looked then at the possible presence of dopaminergic neuronal death in the substantia nigra. And what we could see is that at 9 months of age, there was a tendency in the decrease of dopaminergic neurons. This was measured by stereology, but was not significant. At 12 months, there is a 30% reduction in the dopaminergic neurons. And then at 20 months, there is a 50% of a reduction in dopaminergic neurons. And the bona fide, the fact that this is not only a loss of TH staining, but a real loss of neurons was proved by using UN and counting the neuron in the system. Then we say, okay, could be that the dopamine reduction is due to loss of terminals. We measure the TH neurons, neurites in the striatum, and we could find that there was a slight tendency at 9 months to reduction, but the significant reduction was only at 12 months. So when we have already 30% of neuron loss. However, when we went to look at the behavior, we couldn't find really much motor behavior using rotor rod that is not a very sophisticated sensitive system. Only at 18, 20 months, when you have a 50% loss of neurons, you see a defect. But this is a bit like you can see in patients. 50% loss of neurons is required for manifestation of the motor behavior. And also another test that we used was the static rod walk, and we could see that again, only at 15, 18 months, we have a significant difference in the month. However, if we look at the dopamine release that we observe in the striatum, this is already present. The effect is present at 6 months. This is before the 30% cell loss, before this cell loss, and it is also before the reduction in the significant reduction in terminals, indicating that indeed this loss of dopamine release, it is a functional. It's not structural due to the loss of neurons or loss of terminals, but depends on the system that leads to the release of dopamine. So we look at a system that behavioral system that could tell us more subtle changes that could be related to earlier age in the mice. And what we could find that using DigiGate, that indeed at 9 months of age, you can see there are significant difference in the transgenic mice in the stride length, in the stance length, and the swing duration. These are all small behavioral features that they are also present. They can be present in the patient, but in these mice are very characteristic, and they allow us to detect changes much earlier. So having identified a mouse with cell death, with lots of with dopamine deficit and also motor behavior, what we want to do is to test compounds to see if this was really by rescuing this defect, we could, or the alpha-synuclein aggregation, we could affect behavior. So what we did, we used an ANLE138B, a compound that was given to us by Armin Gies and Christian Griesinger and produced by Andrei Leonov and Sergei Razman in Göttingen. And this compound is an oligomer modifier. So the idea here you see is the structure. It was tested also in other diseases, and this somehow affect the aggregation of the proteins. And then we use that cross the blood-brain barrier. So we put in the food in the mice, and these are the mice after 3 months treatment. So 9 months when we have the beginning, the dopamine release defect is already present, but we have the beginning of cell death, not significant, until 12 months when we have 30% cell death and a very, very pronounced loss of dopamine release. And what we could find is that the drug given in the food just reduce the amount of alpha-synuclein aggregation, but more than removing the aggregates, they are like chipped, so they are not so dense, and there is a large increase of monomeric alpha-synuclein in the system. And these changes in aggregation correspond to a rescue in the dopamine release in the striatum after potassium induction. As you can see here, these are the mice treated with placebo, and this is the synuclein mice treated with ANLE138B, and these are the control mice treated with the same drug or placebo, but also prevented neuronal cell death, dopaminergic neuron cell death. And you can see here the difference between the significant. So we had found a system that allows to test compounds that can prevent the loss of neurons and as well as the disease. We look again at digigate in mice after ANLE138B, and we could find that the majority of the behavior that we had observed is completely rescued, indicating that indeed the defect that we had observed in this fine movement test was associated to the loss of dopamine release that we had found in the mice. The point was, can we really prove that the synapse is involved? And so what we did, we start to look at another protein that has been shown to be involved in a snare complex formation and also in possibly release than neurotransmitter, but in a way different from alpha-synuclein, in a parallel way. And this protein is the cysteine string protein alpha that associate with HCSC70 and in order to form complex, and then it is involved again in the transport of the vesicles for release of neurotransmitters, also through calcium and different pathway compared to synuclein. So we looked at Laura Callo in the lab, look at the presence of CSP alpha, and strangely enough, we couldn't find any change in the total homogeneity in the brain of the mice, in the striatum. But when you look at the synaptic fraction, there was a clear change in the presence of the protein in the transgenic mice. So we decided to look more carefully at that to determine if the CSP alpha expression, if increased, could rescue the deficit in dopamine release observed in the transgenic mice. So we gave CSP alpha increase through injection of AAV virus expressing CSP alpha in the substantia nigra. We could prove that the protein is translocated, is present in the injected, the protein expressed is present in the synapse. And you can see here that indeed this deficit observed in the synuclein mice in dopamine release, it is rescued by the expression of CSP alpha. And how does CSP alpha do this? Again, you can see this is alpha-synuclein presence aggregates in the striatum of these mice, and this is reduced by expression, not overexpression, expression of CSP alpha by viral injection. And so this is the difference you can see in the aggregate number of alpha-synuclein, and this is alpha in the mice, and this is instead is in after CSP alpha treatment. So the number is clearly reduced of the aggregates by treatment with alpha-synuclein. And again, you can see here as this monitored by super resolution microscopy in collaboration with David Kleinerman and Eric Idari. And you can see that these aggregates are much, much smaller. And again, like Kalle, we have a large increase of monomeric alpha-synuclein. So what we think is happening, this alpha-synuclein is involved in movement of vesicles through the snare complex binding to synaptobrevin, to syntaxin, SNAP25, and then this vesicle releases the neurotransmitters. But when alpha-synuclein is clamped, recludes all the other proteins, and therefore there is no possibility of release of neurotransmitters. And therefore what it is possible is that Parkinson's disease could be a dying back mechanism, which means that it's not the cell body where possibly the pathology starts, but it could be at the level of the synapse where we find these clumps, very, very early, these clumps of alpha-synuclein and other snare protein. In this case, the dopamine release with this clump stopped to be released. There is a degeneration of the terminals until you have only the cell body in the substantia nigra that remains and the Lewy body that are present there until the neuron completely die and the Lewy body disappear with it. So I hope to have shown you that the presence of the alpha-synuclein presence in Lewy body in the filaments is not only an epiphenomenon, but alpha-synuclein is clearly involved in the pathological process in diseases where it aggregates and it is present. This, in Parkinson, could happen through an influence in the snare protein and the abnormal release of protein. In synucleinopathies and in transgenic mice, there is clearly the alteration of the synaptic function and the dopamine release, and both treatment with the oligomer modifier or increasing the amount of other chaperone protein can affect the synaptic function of alpha-synuclein, reducing the aggregates and rescuing the effect and increasing monomeric synuclein. And finally, this could indicate that perhaps we shouldn't only look at the Lewy body when trying to determine Parkinson's disease presence, but also look at the synapse. And this in particular for the early intervention to cure the disease at the level when it is still like a REM sleep disorder or so. And I would like now to, this has been a collaboration for many, many years. And with Michel Goddard, we started all this. And I have mentioned several of these people, Armin Giese, Christian, Andrei, and Sergei, who gave us the only 138B, Uri and Dana, with whom we did the first storm super resolution in the MI2 mice, Bernardino Ghetti that has given us always tissue to study, and then David Kleinerman, Eric Idari for this storm present in the new mouse model, and then Arianna Bellucci for the measurement of dopamine and important discussion, Jeff Daly also for the dopamine, and then Professor Burgoyne from Liverpool, who gave us the cDNA for CSP alpha. So this work over the years, the paper we published in 2019, it was about 8-year story because the mice had to be re-derived and to age to 12 and 20 months. So it took really, really long time. But the work was done by people that now are not anymore in the lab, like George Tofaris, that is in Oxford, Pablo Garcia Reitbock in UCL, Oleg Anitik in Leeds, but in particular by Mikal, that has gone back now. Mikal is this high, tall guy here, who has now gone back to Warsaw, where he has set up his own lab. And then by Laura Calo, that is one of the column of the lab, and she knows everything about alpha-synuclein and many techniques. And then Emma Carson also, that is involved very much in the microdialysis. And these are the rest of the group where other people are involved in synuclein studies or in other studies. And thank you very much for your attention.
That's great. Thank you very much, Maria Grazia, for a fantastic seminar. Of course, this system doesn't allow for you to know how much the audience appreciates it, but I can tell you from the questions that have been coming in, they're nearly all starting with what a fantastic talk. I'm sure all of our viewers will be very pleased with the presentation that they've just seen. So we have quite a lot of questions. And I'm going to pick some of them that I've been looking at while they've been coming through. In some cases, I will attribute them to the question asker. In some cases, I won't, where they've been asked by clearly more than one person. So this question is from Tom Trainer, who asks about soluble alpha-synuclein, that this can be elevated sometimes in Parkinson's disease, and what role do you think that might have?
Yeah, I think that the soluble form elevated has to have a trigger to start to aggregate. We know that some polymorphism in the alpha-synuclein gene can induce a normal increase in the expression of the protein. So some people, they have more, but probably what you need is a trigger that starts the beginning of the aggregation that then promote everything else. The thing is that what we saw with the treatment that we used, in most cases, we have an increase of soluble alpha-synuclein. So clearly, probably this treatment prevent the aggregation, but maybe they also make the protein more suitable for digestion.
And then a related question from Suzanne Schneider is about the treatments targeting the aggregates in dementia have been largely discouraging. And you know, she asks, what's your opinion about what the best targeting approaches might be going forward?
Yeah, what we have seen at the moment is that there are many different approaches, mainly for the synucleinopathy, but also for tau. And so these two compounds, the drugs that, I mean, it's more difficult to give CSP alpha because you need viruses and so on. But for example, the oligomer modifiers affect the aggregates, so they are reduced in consistency. And clearly that is beneficial. This compound is now, has been, MODAC has developed now for trials, and the trial has just started at the beginning of the year. Now, of course, has been halted, but it looks very promising. What these different approaches all aim is to reduce the amount of synuclein when you have aggregates. Because of course, not having very, very early biomarkers of the people that will develop the aggregates, we can intervene only when something shows that there are aggregates. And in this case, both by intervening, by reducing the transport or the amount of monomeric synuclein or small aggregates with antibody, or just other people, sponsored also by the Michael J. Fox, they are reducing the amount of alpha-synuclein by siRNA. And I'm sorry, I forgot, the Michael J. Fox also sponsored this work, our work. And so there are many different approaches, which seems to all be direct to reduce the aggregates. And then if there is the aggregates that are toxic, and therefore there is a gain of toxic function, or the beneficial effect is because there is more monomeric synuclein available, and therefore they could also imply some sort of loss of function. We really don't know that. And that is something what everybody tries to understand.
Thank you. So Jessica Christini Breedy asks if you have any idea how alpha-synuclein might cause alteration in the localization of other synaptic proteins such as VAMP2.
Because alpha-synuclein binds, Sudoff has reported that binds to VAMP2, and VAMP2 is a membrane protein that is involved in the snare complex formation and interacts with syntaxin, with NAP25. Therefore, it is possible that this binding of synuclein with VAMP2 then triggers the aggregation of also all the other proteins that in theory should have their organization, and that they should have a very specific distribution at the synapse. So disrupting one could be like a phenomenon that leads to the disruption of the other.
Okay, thank you. And a question from Jessica Kwok regards the transport of alpha-synuclein between the gut and the brain and between brain cells. And does it mean that alpha-synuclein filaments or inclusions can be found in the extracellular matrix?
So far, we have not found aggregates in the mice, at least in the extracellular matrix. They are mainly intracellular. And sometimes if we have the impression that there are, like, for example, also in human brain, Lewy body that seem extracellular, if you look more carefully, there is always an axon or a dendrites that is very much enlarged. So to my knowledge, we know that it has to go through the exosome, nanotubes, release of alpha-synuclein in some form of vesicles, but to my knowledge, there isn't a great literature on the aggregation of alpha-synuclein extracellularly. It happens inside the cells.
Yeah, and then a question from Elisa Manozzi is what do you think about the pathogenic role of Lewy bodies, thinking about the relative lack of Lewy bodies seen in the LRRK2 related Parkinsonism?
Well, LRRK2, it has also other functions, so it cannot rely only in the Lewy body, although there are Lewy body, but LRRK2, for example, can give also tau aggregation instead of alpha-synuclein aggregation. And it has been, some cases have been found to have tau aggregates and have progressive supranuclear palsy. So I think that one should look more at the pure cases of sporadic cases or cases where there are modification with alpha-synuclein, like mutations or duplication or triplication of the alpha-synuclein gene. And what I think is in this case is the protein that probably reach a level in which the first seeds of aggregation are formed. And therefore, you have the aggregates. However, I am not sure that, as I mentioned earlier, the Lewy body are really the first thing to look at. So some people say, oh, incidental Lewy body disease. There is no disease, but there are Lewy body in the brain. But how many Lewy bodies are in the brain? How many neurons have died? And so how many synapses have been lost? So just to look, for example, the presence of 10 Lewy body or 5 Lewy body in a field without any cell death, I think it's a very early stage that perhaps is not the one where one should look at the body as the cause of the disease, but look from our results, for example, at the synapse, and it is an evolution that will lead then to the worsening of the disease when the cells start to die. But certainly they will start to die in relation also to the presence of the Lewy body, because when you have these aggregates of filaments that can trap and may be an obstacle for the movement of a protein in the cytoplasm, it is very unlikely that the cells will be very happy, because there are chaperone deficit, because they bind there. The proteasome and the autophagy are inhibited by these large aggregates. And so it is possible. We still don't know how the cells die, but it has to be related with the presence of the aggregates. But I'm not 100% sure that should be the first thing to look at.
Great, thank you very much. So I'm afraid we don't have any more time for any further questions before our allotted time. So I apologize to those whose questions that I've not been able to ask. It remains for me to thank Maria Grazia Spillantini for a fantastic Thudichum Medal lecture and congratulations on being awarded the Biochemical Society Award. for this year. So we welcome suggestions for future topics and speakers to feature in the Biochemistry Focus webinar series. So if you've got any ideas for a webinar, please submit a proposal on the Biochemical Society's website. Next week, on the 16th of July, we'll hear from Dr. Sara Priego Marino from the Salk Institute for Biological Studies for her Biochemical Society Early Career Award presentation on disassembly of the replicative helicase and eukaryotic cells. So I hope you'll come and join us for next week's webinar also. And that remains for us to just thank Maria Grazia once more for a fantastic seminar. Thank you.
Thank you very much, and thank you, Biochemical Society, for the medal. The medal is a really a great honor. Thank you, and everybody for the attention.